Hello everyone. So today I will proceed with the fertilization process, but at first let me just give a brief idea about the hormonal regulation of menstrual cycle. So in the last video I discussed about how the hormones are regulated during oogenesis. So the same hormones, that is FSH, LH, estrogen and progesterone, these are regulating the menstrual cycle in females as well. So in case of the anterior pituitary hormones, we have two hormones, FSH and LH. So as you can see that when the follicles are in growing stage, then the estrogen is taking a peak, because we have already seen in the last video that estrogen is responsible for the growth of the follicles and the maturation of the follicles. And when it reaches the ovulation stage, LH is taking a sharp peak, which is known as the LH surge. And this LH surge is responsible for rupturing the ovarian wall and releasing of the ovum from the ovary. So here you can see that the follicle stimulating hormone is also taking a short peak. But in case of LH, it is a sharp as well as a very much increase in the concentration of LH. So this is known as LH surge, which triggers the ovulation of the in the menstrual cycle. So this ovulation occurs in the 14th day of the menstrual cycle, and from here the luteal phase begins. Luteal phase means the corpus luteum is generated. So after the ovulation, the Graafian follicle is converted to a corpus luteum, and therefore here the progesterone is taking a is rising in levels, because the progesterone is responsible for maintaining the corpus luteum. The progesterone is responsible for maintaining the thick endometrium wall of the uterus, and therefore the progesterone is increasing rapidly. So here you can see that the LH and the FSH are taking a dip, like they have decreased in the concentration and the levels have decreased so much that they are almost in the parallel with the previous or pre-ovulation phase. So here they are taking a dip, and estrogen and progesterone are working. So progesterone is working for the endometrial lining as well as the maintaining the corpus luteum. And here you will see, if the fertilization has not occurred, then the hormone which is known as HCG, human gonadotropin, chorionic gonadotropin, that hormone is not released from here. And if it is not released, then corpus luteum degenerates, and therefore progesterone is also decreasing. And here the progesterone, since in the absence of progesterone the uterus cannot maintain such a thick lining, so the endometrium is falling apart and it is being shedded off, and this for this reason the menses are happening every month. Like it is bleeding out, and therefore for day 1 to 5 the menses are occurring. This is after the 28 days. And after that the follicular phase again begins, and the same thing, the follicles start growing until one reaches a maturation stage, and then ovulation occurs at the 14th day. So this is the hormonal regulation of menstrual cycle. And this is the basic structure of the Graafian follicle. So as you can see that the Graafian follicle is covered by two cellular layers. One is the theca externa, and the layer just beneath that is the theca interna. Next is a thick layer, or two to three layers of granulosa cells. And from here the granulosa cells are getting specialized into the cumulus oophorus cells, and this is a region of specialized epithelial cells, and this is known as hillock, where the ovum is eccentrically located. Like it is not exactly in the central part, but it is located in the eccentric part. And this is the basically the secondary oocyte that is being formed, and it is covered by a vitelline membrane. And this space between the plasma membrane and the vitelline membrane is known as a perivitelline space. And next is a layer of zona pellucida. This zona pellucida is a non-cellular layer, and it is it is a very thick covering, but it is non-cellular. And next is a layer of cellular layer, which is known as corona radiata. And these layers are basically here for protection of the oocyte or the ovum. And here you can see that this entire structure is then covered by cumulosuphorous cells, which are nothing but the follicular cells, modified follicular cells basically, and they are all responsible for protecting the ovum. So these are the layers of an oocyte or a Graafian follicle. Next is the mature ovum. This is the mature ovum. As you can see that this ovum, after ovulation, it is only valid for 24 hours. Like, it can be fertilized by a sperm only within 24 hours. After that, it loses its ability to be fertilized. So in the ovum, there is corona radiata, zona pellucida, like I said earlier, and then vitelline membrane. And then the nucleus is specialized and it becomes a large nucleus. It is known as germinous vesicle. And the cytoplasm of the ovum is known as ooplasm. And there is another important term which is known as the polarity. So as you can see that the region from where the polar bodies are being extruded from the oocyte, that region is known as the animal pole. And the pole opposite to that is known as the vegetal pole. Usually the vegetal pole contains the maximum concentration of yolk in the egg. But in case of human eggs, yolk is absent. So the pole opposite to the polar bodies is known as the vegetal pole, and the pole from which the polar bodies are being extruded from the oocyte, it is known as animal pole. So then moving on with fertilization. So fertilization is basically the union of two haploid gametes, male gamete, sperm, and the female gamete, egg, and it restores the diploid condition of an organism. So the zygote is basically a 2N cell, and therefore the diploid condition is restored. And hereby the zygote, from the zygote, a series of mitotic cell division starts, and it results in differentiation, like I had said earlier, organogenesis, and finally the embryo development starts happening. So here you have, as you can see that the sperm has entered from here, and it goes on up to this portion, the fallopian tube, and here the fertilization actually occurs. So from the ovary, the egg has released by the rupture of the ovarian wall, and it is entering the fallopian tube here. So here, if the fertilization has occurred, then it will form 2 cells by mitotic division, then 4 cells, then 8 cells, and it will move on like that. And then it will again reach the uterus, and here it will attach with the thick endometrial lining of the uterus. This process is known as implantation. So if this implantation has not occurred, then the menstrual cycle will shed off the endometrial lining and the menses will begin every month. So this process is very important, and here when the implantation has occurred, then the endometrial lining will be retained and it will be maintained in this thick condition. So fertilization is a very complex process, and it includes a lot of events. So the 4 major events that are there, I will begin with the sperm and egg contact and recognition, and to ensure that species specificity is there. Like, how will they know that the sperm and egg are of the same species, and how do they recognize each other, how do they come in contact, and so on. Next is the regulation of sperm entry and allowing only one sperm, like only one sperm will be allowed to enter the ovum and fertilize it. More than one sperm will not be allowed, and this process is known as prevention of polyspermy. Next, the gametic material of both the gametes, the genetic material like the N and N, the haploid set of chromosomes will fuse together, which is known as karyogamy. That is, the nuclei will fuse together and it will form the diploid set or 2N set of chromosomes. And the last one is the activation of egg metabolism to begin the development process. So it will act, so all these events will lead on to egg activation, and finally the metabolism will start, the respiration and everything will start, and the embryo will be developed. Okay, so the first process is known as capacitation of sperm. So capacitation is basically the series of changes which a sperm has to undergo, because otherwise it will not have the ability to fertilize an egg. So this capacitation occurs in two events, like the fast event and the slow events. So at first what happens is that there is the removal of decapacitation factors from the sperm head. So as you can see that the sperm head has a lot of these factors. These factors are nothing but certain surface molecules of the sperm, and with that the seminal plasma proteins have coated on the surface molecules of the spermatozoa. So all these has to be removed, and the sperm surface molecules has to be exposed properly so that it can bind with the egg. So this is done by the whenever the sperm reaches the vaginal environment, it is done there. After that, the seminal fluid helps in neutralizing the acidity of the vaginal fluid, and this alkalinity also helps in the increasing the activity of the sperms. So as you can see, once these factors are removed, then the sperm membrane changes. So this cell membrane loses its cholesterol, increases the membrane flexibility, and the calcium influx occurs. So all these results in the flagellar movement, like in vigorous flagellar movement starts occurring, and therefore the sperm increases in motility, increases its motility. This is known as hyperactivated motility. So these are the slow changes which occur after 1 hour of the sperm entering the vagina, which is known as insemination. So these are the slow changes that are occurring. Glycolysis is increasing, obviously, because it will require a lot of ATP, and therefore this flagella starts moving asymmetrically or in a vigorous movement. So here also you can see that when the sperm was in epididymis, the surface receptors were were exposed, and then the seminal plasma proteins bound with it when it was ejaculated. So after entering the female tract, here you can see that it has been capacitated, which means it is losing the seminal proteins which were coating on the surface, as well as it is losing some of its receptors also. So the fast changes occur like within 10 minutes of the sperm entry, which is the membrane fluidity is increased by, of course, by cholesterol removal, and then lipid raft aggregation, as well as here the fibronectin expression is increased. Fibronectin helps in adhesion of the cell surface, so it will go and bind with the egg more properly now. And also docking of acrosome. So acrosome also starts docking. Acrosome, like I mentioned earlier, is the head cap of the sperm. So all these changes are known as sperm capacitation, which helps in ultimately enhancing the activity of the sperms and leading to the proper fertilization. So all these are the sperm changes which are involved in sperm capacitation, like calcium influx occurs, cholesterol efflux occurs. Efflux means going out. Then increase in protein tyrosine phosphorylation. Also the intracellular pH is increasing. Bicarbonate ions are also incoming, and hyperpolarization of the plasma membrane, as well as increasing in CAMP and ROS levels. CAMP is the cyclic adenosine monophosphate, which is a second messenger, useful in a lot of signaling pathways. And here you can see that in the fast events, the sperm motility activation is there by, of course, by sodium and bicarbonate. They are this is the co-transporter, sodium bicarbonate co-transporter. It is incoming inside the cell. The caspar is a specialized sperm calcium channel which is helping in calcium influx. and all these are helping in the soluble adenylyl cyclase enzyme to generate more and more CAMP, which is further going to activate this pKa, which is the protein kinase A. And from here the phosphorylation of pKa substrate starts. So all this leads to the sperm motility activation. The pH is also increasing because bicarbonates are getting inside. And in case of the slow events, the cholesterol efflux is a very distinct thing. Cholesterol efflux will happen, BSA, that is albumin will help in the efflux of cholesterol from the plasma membrane. And of course it will lead to increase in protein tyrosine phosphorylation, sperm motility hyperactivated, and preparation for the acrosome reaction. So all these changes are leading to the acrosome reaction, which we will go to next. In the acrosome reaction, as I have already mentioned, that acrosome is the cap part of the sperm. So as you can see, when it is coming in contact with the zona pellucida of the egg, then calcium influx starts occurring. So acrosome contains a lot of enzymes inside it, and thereby when it reaches the zona pellucida, it reacts with it. It starts releasing its contents here, which helps in its penetration inside the egg. So acrosome then reacts with the perivitelline space. Here there are protein receptors which are responsible for binding with the sperm head, and therefore it destroys all the layers and the receptors while going inside the oocyte. So here also you can see that membrane depolarization has occurred, and depolarization of the membrane is an important step for preventing polyspermy. It will not allow other sperms to enter inside once one sperm has been allowed. So the sperm nucleus, then the sperm plasma membrane fuses with the plasma membrane of the egg, and the nucleus is released inside the egg. So where it binds with the, where it goes and fuses with the egg nucleus, which is already present inside. So as you can see that I have listed the contents of the acrosome, like they are collectively known as sperm lysines. So what are these? These are nothing but certain enzymes or which help in the digestion of the proteins which are present in the layers of the eggs. Like hyaluronase is responsible for the digesting the ground substance of follicle cells. Corona penetrating enzyme responsible for the corona radiata destruction. Zona lysine or acrosine responsible for zona pellucida digestion. So all these are responsible for digesting the layers of the egg. So here also you can see that with acrosomal reaction, the acrosomal enzymes are coming out, and this is the actin center, that is the globular actin, which is getting transformed into F-actin, filamentous actin, by actin polymerization, and this actin is protruding and becoming a process which is known as acrosomal process. And in the acrosomal process, there are certain proteins called bindin, which are responsible for species-specific recognition of the egg and sperm. So this bindin is very specific for each species of organism. So it will recognize, the egg has receptors which will recognize the bindin of that species, of the sperm of only that species. So here the species specificity is maintained and the binding occurs like that. So here you can see there are a lot of granules or vesicles beneath the plasma membrane of the egg, which is known as cortical granules. So these cortical granules are vesicles containing certain enzymes and proteins, which after the sperm nucleus has entered in the egg cytoplasm, they start releasing their contents in the perivitelline space, that is with between the vitelline membrane and the plasma zona pellucida. So this process is known as cortical reaction, which we will move on to next. Okay, so this cortical reaction is basically the cortical granules, which have the enzymes, they are releasing their contents in the perivitelline space, which helps in the detachment of the vitelline envelope, and also they help in the modification of the sperm binding proteins of the egg, and therefore it helps in the prevention of polyspermy. Also, it helps in the osmotic swelling of the envelope, and it makes the envelope go away from the egg membrane. Therefore, the connection between the membrane and the envelope, which was through certain polar like poles, protein poles, that anchoring is also destroyed and it moves away. So all this is done to prevent the polyspermy, basically, so that other sperms cannot come and enter the egg once one sperm has been allowed and one sperm nucleus has entered the egg. Other sperms should not be allowed anymore. So these cortical granules, they release the contents in the perivitelline space. The contents include proteases, hyaluronic acid, peroxides, and hyalin. So all these are responsible for prevention of polyspermy in some way or the other. Like proteases, they help in the detachment, they help in the breaking down of the protein poles, thus detaching the envelope from the plasma membrane, so that it goes away, and the sperm binding receptors are much away from the vitelline envelope. Therefore, this sperms cannot come and enter here. Hyaluronic acid is also responsible for making this region. If hyaluronic acid is released, then it will absorb a lot of water and start swelling. This envelope will start swelling, and the peroxides are responsible for hardening of this envelope. Therefore, the sperm cannot easily enter through it. And hyalin is of course responsible for creating another membrane, which will be further known as the hyalin layer later on. So here also you can see that the cortical granules are here, the plasma membrane is here, and these are the sperm binding proteins. So what will happen is that if these cortical granules release their contents into the space here, then it will start hardening further and further, and the other sperms cannot come and enter easily through it. Also, the attachment size, like the binding proteins, are much further lower down here. So what will happen, these sperms will not come in easily in contact with the attachment sites. So all this will lead to prevention of polyspermy. So this sperm entry, as I have already said, that it triggers IP3 mediated calcium release from the intracellular stores of the cell, and this calcium release, the increase in overall calcium of the cell cytoplasm, this leads to the fusion of the cortical vesicle membrane with the plasma membrane of the egg, and therefore they start releasing their enzymes in the perivitelline space. So this is the egg. Basically, there are a few glycoproteins in the zona pellucida layer of the egg, and these glycoproteins are termed as ZP1 to 4. So ZP1, 4 and 3, they are responsible for primary sperm binding. ZP1 function is not known, is not clearly known till now, but ZP3 and ZP4 can bind the capacitated sperms easily. So they are the receptors for sperm binding basically. And after acrosomal reaction, like acrosome reacted sperm can bind with ZP2. So this is known as secondary sperm binding. So after the release of the cortical granules contents in the space, perivitelline space, these receptors or these glycoproteins are modified such that they cannot bind with the sperms any further. So that is also another reason why polyspermy is prevented by cortical reaction. So here you can see that the cortical, this is the fast block to polyspermy is by depolarization. Membrane depolarization occurs when there is an influx of sodium ions, calcium ions, etc. So membrane potential at resting stage is at -70 millivolt, and it increases to +20, +30, up to +40 millivolt, and it becomes depolarized. After that, again, potassium ions efflux occurs, and therefore the potential drops down to the resting stage. This is known as repolarization. And if the potassium channels keep or like if they remain open, then the potential will drop down further to hyperpolarization state, like lower than the potential of the resting state. And again, after some time, when the potassium channels are closing and the sodium channels are opening again, then it will reach to the resting state. So this is known as depolarization of the membrane. And this depolarization, when it has occurred, it prevents the sperms to bind or come closer to the egg. So what will happen is that sperms will not come easily and bind with the egg. And therefore it will take some time that whenever the sperm entry has occurred, when the sperm egg fusion has occurred, then it will prevent the other sperms from reacting with the egg membrane by depolarizing the membrane. And therefore it gives some time to help in the acrosomal reaction and the cortical reaction to occur. And after the cortical reaction has occurred, this will finally be the slow block to polyspermy, which is the envelope, the vitelline membrane will shift away from the cell membrane. It will create a space here which will be filled with fluids, mainly water. It will start swelling away, and this will harden, and after hardening it will be known as the fertilization envelope. So this fertilization envelope prevents other sperms from entering into the egg. So this sperm can then successfully release its nucleus inside the egg cytoplasm, and therefore the egg cytoplasm, which already contains the egg nucleus, it will fuse with it and karyogamy will occur. That is the fusion of the nuclei, both the nuclei. So this is known as a fertilization cone. When the plasma membrane of the egg has fused with the plasma membrane of the sperm, then the egg cytoplasm starts to engulf the sperm head and the sperm nucleus inside. And this formation is like an inverted cone. This gives the shape of an inverted cone, and therefore it is known as a fertilization cone. So up to this, the nucleus is released now, and therefore the next we will move on to zygote formation in the next video. Thank you.